Hi, everybody. My name is Melo, and I'm a PhD student at UMass Amherst. I'm working with Hawk Mountain Sanctuary on their American Kestrel project. So, pop quiz. Can anyone name this bird for me? Anyone? 10 points? Yes, this is a male American kestrel. They're beautiful, but also they have some really cool characteristics that make them perfect as a model organism. So first of all, they're a raptor, which puts them at the top of the food chain, which makes sure that we can learn about the prey species that they're relying on in order to see how they're doing as a population as well. They're also part of the falcon family, which makes them super cool, but also allows us to use them as a proxy for other falcons that we might not see as often, like merlins and gyrfalcons. And finally, they use nest box like this dandy right here, which allows us to really look under the hood of breeding biology and see how chicks are developing, how reproductive success is year after year, and maintain nest box programs, like I'm sure a couple of you guys do, to see these long-term trends. So further, of course, these nest boxes have to be placed somewhere. So where are American kestrels found? You can see from the range map, they're everywhere, which is really amazing. So that means they're super adaptable to a bunch of different environmental conditions. We can see they breed all the way up into Alaska and winter and spend sometimes all year round, all the way down to Central America and South America, which is pretty awesome. And further, within these different areas, they're usually found in farmlands and meadow type habitats. So as you guys, I'm sure are aware, warehouses are kind of encroaching on this. So they're starting to become an issue that we're thinking about when we're thinking about kestrels needing a place to live. But it's not just warehouses. We also have to think about what kestrels are eating. So like we said, they're at the top of the food chain. So they're going to be eating a couple of different specific things that we're going to keep an eye on. So first, they're eating insects like orthopterans or grasshoppers. They're also eating small songbirds that are typically about European starling size or smaller, as well as small mammals such as meadow voles. So with all these different food types, it not only makes them super adaptable to live in these different areas, it also gives them this really cool trend as a farmer's best friend. So all of these different prey species that they're eating, such as rodents, are all going to be agricultural pests. So rodents are going to consume and contaminate grain. They're going to girdle orchard trees and other large plants. And the same thing with songbirds and insects. They're all going to damage plants or cause an issue in some way in conventional agriculture. So instead of putting out chemicals, we can employ kestrels by putting out kestrel boxes on barns in order to have a natural pest control system that kestrels are willing to do for free. So it saves farmers money, keeps chemicals out of the environment. So unfortunately, since conventional agriculture is really found throughout the United States and further, we have to think about not everyone is going to be using kestrels, right? So eating all these different prey items leaves kestrels vulnerable to conventional agricultural chemicals. So thinking about the three things that they're going to eat, the rodents are likely going to be contaminated with rodenticides. This isn't just agriculture. This is us personally as landowners putting out those little rodent bait blocks, those anticoagulant rodenticides. Further, insects such as grasshoppers can be contaminated with insecticides. You know, farmers are spraying fields or they're putting out neonicotinoids, which are also going to change or contaminate songbirds that are granivorous, such as some of the birds that the kestrels are going to eat. So all these different vectors or ways that kestrels are going to get contaminated really leave them vulnerable to population declines over time, which is really the thought about this talk. So we know that contaminants may not be the only thing that we can blame here. Here we go, clicking. There we go. We're seeing that the population isn't doing very well. Since the 1970s, kestrels have been declining about 30% in many of the regions that we've been studying. So this graph is using nest box programs. So we can see in the Northeast, that dark downward arrow means it's a significant decline. We can see all of the dark arrows or significant trends are all downward, which is pretty sad. The only place that we have upward trends where kestrels are doing well are non-significant and they're very marginal increases in populations. Looking further into our hawk watch sites, our hawk counts for migration counts, we see a very similar trend. So kestrels are declining. Again, black arrows being significant. They're declining a ton in the Northeast as well as over in the West, and all of our significant trends are downward. We have a couple upward down there in Texas, but again, marginal not really a population increase that we can count on. So what did we do? So Hawk Mountain saw that, this is happening since the 1970s. All of the researchers out there, thank you, are trying to figure it out, but no one's really putting together a comprehensive project to figure out what's really going on with kestrels. It could be habitat change, it could be contaminants, it can be a whole host of things, but no one's really put it all together. So the first thing we wanted to do was to determine the causes of the decline, or some potential causes of decline that we could investigate further. Once we came up with a list, we wanted to investigate how these drivers of decline impact the kestrels themselves, either reproductive success, survival, other metrics that we can measure using nest boxes, as well as telemetry. Once we were able to build these methods, we also wanted to replicate this across the Northeast, where we know kestrels really are not doing well, and eventually spread out west, where kestrels also don't seem to be doing as well as they should be. And finally, we want to develop and communicate management plans for folks like the Game Commission, or other people like landowners that can just implement these programs in order to help kestrels on a local scale that can build up to a regional and global scale. So what could be causing an ongoing decline in such an adaptable raptor? Like we talked about, there's a couple different culprits. So environmental contaminants, like we said, are introduced in multiple different ways to kestrels, and they can really cause a bunch of issues, anywhere from neurocognitive issues, so kestrels aren't able to fly or use their brains as well as they should be, all the way down to just overdosing of a chemical and mortality directly. Habitat change, of course, is also something we're thinking about. Intraguild predation, so like Cooper's hawks or other large raptors eating kestrels, since many of these other raptors are increasing in populations, there is some evidence showing that they're eating kestrels at a higher rate than previous. So we're thinking about that, as well as decreasing prey abundance and all of these other things. So there's tons of things that could be happening to kestrels. But for the sake of our study, we chose to go with we decided to go with those four main ones here that we've been chatting about the most. But the thing to think about with those different factors is that they all might be interacting. So I asked you guys to walk through me quick from the transportation of a large meadow habitat, which would be ideal for kestrels. They have plenty of prey, plenty of perch substrate. They're able to hunt wherever they want. And then that being developed into a residential development. We're seeing this more often than not, unfortunately. And it's causing habitat change to directly impact kestrels. They don't have a place to live. They don't have a place to hunt. But further, when we develop into residential areas, we're also introducing contaminants, right? And those contaminants not only can be harming kestrels directly, they're aimed at reducing their prey. So we're putting out rodenticides not only to kill rodents, which is going to decrease prey, but it also might impact the kestrel themselves if they ingest it secondarily. Since there's less prey, now they're interacting with their competitors. They're all competing over less prey and fewer resources, which is eventually going to cause more, they're going to be competing and fighting with each other for resources. So not only do they have a worse place to live, that place has less food, the food's contaminated, and they're competing for less. So what we did for this project is we determined four main drivers of decline that we wanted to investigate. So environmental contaminants, this decreasing prey abundance, habitat and land cover changes, and the increase in Cooper's hawk populations over time. And we also took data on the ecological responses of kestrels that might be correlated with these different drivers. So we looked at nest box occupancy, nestling growth and development throughout that nestling period, fledgling survival and dispersal using transmitters, as well as adult seasonal survival. We tracked adult kestrels year round to see what's going on. So by taking all of this data all at the same time, we can see how contaminants impact nestling growth and development. But we can also see how decreasing prey abundance might impact nest occupancy. And literally every other interaction of what driver impacts what ecological response. You can just put 2 and 2 together and see what's really happening with kestrels. So what does this look like in the field? It looks super fun. So the first thing we do is we use these nest boxes to assess nest box occupancy. So luckily the nest boxes have a door, so you can just open her up and see. We can see if there's kestrel. We can see how many chicks, if the chicks survive to fledging date, and how they're doing overall. Once we see a box is occupied, we'll measure those chicks. This little guy, 14 days old, we'll measure mass as well as different wing measurements, tail measurements. pretty much everything you can measure about growth and development. Once the chicks are old enough, I'll deploy transmitters and color bands. You see our transmitters are super small little guys. They're less than 3 grams, and these allow us to track them for up to 8 months after they fledge the nest. We're also collecting blood samples at this point to look for environmental contaminants. And we're surveying prey populations at the same exact time at the same box. So using this technology I developed out of downspout gutter, we can look at small mammal populations by putting ink on these ink pads and bait in the middle. So we can see when the mammals walk across the ink, they leave their tracks, and we can assess how many mammals there are in the area. Similarly, I didn't bring the net, but you can see we're sweeping set transects to look at what kind of insects are going around and what abundance of insects we have around our nest boxes. We're also doing songbird point counts, but I'm not good at taking pictures of birds. And the final thing we're doing is a playback experiment. This is something really fun that I developed using a taxidermy Cooper swamp and playing calls. This is an capture of our setup. And we're seeing that kestrels are responding pretty intensely to Cooper hawks being around the nest. And it seems like they're responding more when they have nestlings that are just about to fledge. So we have some super interesting results. We're also looking at some habitat change characteristics on a GIS scale, but that's a little boring. But the very cool thing about this is we're doing this all at the same time at the same exact nest boxes every year. So for the past 2 years, we have all this data on all of the chicks. And not only are we doing it just at Hawk Mountain, we've made a collaboration or a collaborative network of 20 different universities, nonprofits, government organizations all across the East, starting, of course, with Hawk Mountain. Throughout Pennsylvania, we have a bunch of different sites, working with the Game Commission, some other volunteer groups and universities. We're now in New Jersey, Delaware, Virginia, New York, Vermont, Massachusetts, and now we're just moving west to Boise State, Idaho to see if we can get some comparative data out there. We're as far south as Cape Coral, Florida now. And now we've just moved up. We went international. We went to Canada. So we have some associates up there at a consulting firm and at a university that we're able to work with. And all of them are replicating our exact methods, which makes it really great that we can compare their survival because they follow the same transmitters at the same interval as us and compare to see how are things changing regionally. You know, our kestrel's affected by contaminants in Pennsylvania, but not so much in Virginia. And we can really kind of tease apart these different interactions. So a little basis of our timeline. For the past 2 years, we've been collecting field data and samples. We've analyzed those samples so far from other sites and from other labs. And right now, we're in this fun stage of analyzing the data using R and other statistical packages and modeling the relationships. So I'm a PhD student, like I said, so this is the fun stuff of me sitting at my laptop. But it's the important part of putting things together and teasing apart these different trends in order for us to see what's really going on. So once we're done analyzing data, we're currently working on publishing some data. And from there, we'll be developing some management strategies to share with folks to implement on their own land or to implement on a larger scale if it's the Game Commission or other large land-owning organizations. So how can you guys help as banders? So we've been putting out these transmitters and color bands. You can see one of our guys there sporting both. If you ever see a bird with a transmitter or a color band, report it to me because that's probably one of my birds and I can include that in a survival model and say, hey, bird J2 was now found in Florida when I banded him up here in Pennsylvania. Even though you don't have to recapture the bird to read the USGS number, just seeing or having a picture of that color band or transmitter helps me out a whole bunch. Also, if you're ever in a place that you find a dead kestrel, a lot of folks with kestrel nest box programs know that you can find one every once in a while in a nest box. If you find one in a box on the side of the road, wherever, if you can email me, that'd be great. We're running necropsies on them to see why they died, what their contaminant load is like, if they're banded, where they came from, and things like that to give us a better idea of where mortality is coming from. And finally, as landowners or as people that have a stake in, landowning organizations, maintaining good habitat for kestrels, even though we don't have the data to say exactly how it helps, we're sure it helps. So maintaining good habitat is always good for our little kestrel friends. So that's it. I'd like to thank everyone from our collaborative sites. We have a couple of them here today, which is great. And all of our donors and supporters, of course. But yeah, I'm here for questions.
I just have a question about where you should get a Kestrel box.
Yeah, so this one's specifically from Hawk Mountain's Farmland Raptor project. So we build these, and by that I mean I build these. Purchase them. I believe they're around $25, or on Hawk Mountain's website we have plans to make that. So we have
Contact Hawk Island,
Hawk Mountain. Yep, and I have brochures up here as well for our Adopt the Kestrel box. Yep. Cool. Anyone else?
Are you determining foraging areas for your nest birds?
Yeah, so we have just radio transmitters, so we only check them once a week. So we don't have really high quality habitat use data. But one of our collaborators in Virginia puts out data loggers, so he knows exactly where the birds are and is studying that exact thing. If we had higher resolution movement data, we would. But in general, we're using the transmitter's locations somewhat to look at the home range around the nest box in order to look at land use within that range. Yeah. Yes.
I'm curious if you have any data or information about possible interactions at some solar projects. If those are going in a lot of them that don't have it.
Yeah, that's a really great point. I'm glad you brought that up. One of my lab mates is actually looking at passerines in solar fields, so that would be a really good collaboration. I don't have any data on that, and I don't think there's much published on it yet, but definitely an area of concern for sure. Thank you.
That was great. Thank you so much.
Thank you.
And Lindsay, she is in the advancing program manager at Powdermill Nature Research Center, our PhD student at University of Toledo, and our newest third vice president. So congratulations. And she will be telling us about bird observatories in the 21st century.
This works, right?
Yeah, that's
Yeah.
And then you go these right here if you want.
Okay. All right, thanks everyone. I'm going to just talk a little bit about our banding program and the history of our Avian Research Center. So Powdermill, the bird banding program was started in 1961 by a man named Bob Leverman. A lot of you probably remember him. He developed this station with no building, right? He was banding outside. The purpose was just to inventory and study the local bird life just in our local area. But it was, we all know these birds are moving, and so we can expand on that first vision. We have over 825,000 banding records in our data set. It's about 190 species, and we band about 13 to 15,000 birds a year. We process that many a year. So we really progressed beyond just a banding station to an avian research center where we have a lot of projects going on. And I'm not going to sit here and talk about, or stand here and talk about all of these projects that we have, but we do major collaborations. We do a lot with MODIS. We do avian perception of glass, which is a project with the American Bird Conservancy. We had something to do with the second Pennsylvania Breeding Bird Atlas. And everybody remember, the third one is coming up next year, so get your binoculars ready. We had a bioacoustics lab. We had a major Louisiana water thrush project as well. So with the banding station, you can do all of these other projects. In this past fall, we opened this new banding station. So I'm going to talk a little bit about what we can do with this station and invite everyone to please come collaborate with us.
Just mic a little bit. Just to,
Yeah, okay, I felt like it wasn't really picking it up. All right. Okay. So the purpose of this was to, our program was expanding, but our space was so small. So we now have a bigger space. We can become this hub for scientific research and collaborations, but we can also expand our outreach efforts as well. So here's our new research space. We haven't taken a lot of pictures inside yet, but this is what it looks like. So in that top picture there, that's what the old banding room looked like. It was a cinder block building. It was fine for the banders, but if any groups showed up or any visiting research showed up, it was so cramped. And so we've been able to replicate that room where the main banding research projects are going on, but there's now more space as well for visitors. We also have a new lab and office space. So this is like a lab bench where we can do things like centrifuging blood, and we have computer stations for the field techs or visiting researchers. So with this new space, we can sample, we regularly sample these physical things like blood, feathers, and fecal samples. We can attach these samples to a banding record, meaning now we know the age and sex of that bird. We have the measurements, but we also know something else about the birds. We can study things like diet, whether birds have microplastics in their systems. We can look at maybe stable isotopes, disease and parasites, genetics. All of these things we can do with just a few samples. These things are very quick and easy to do, especially if we have the researchers themselves come on site, or maybe we just do it in-house as well. We can also collect ectoparasites. So ticks are pretty awful. I don't know how prevalent they are at everyone else's stations. This is something that was really never there until maybe 15-ish years ago. We're seeing ticks a lot more often at our stations, and that Canada warbler is not necessarily an exception. We see that. It's pretty terrible, but we can collect these ticks. We can see if they're carrying diseases. We can collect blood samples from the birds. We can see if the birds are also carrying the diseases, and we can learn something about transmission. We have an entomologist on staff who really loves hippoboscid flies. So if we can, we'll collect the hippoboscid flies. She loves them. She loves the parasites that the hippoboscid flies have themselves. And there's other ectoparasites too that you can collect. And so we have visiting researchers come. So this is a project. This is a PhD candidate. She's defending her thesis on Monday, or her dissertation on Monday. So not a candidate for much longer. Claire Nemes is studying the effects of, so the non-lethal effects of potential predators on birds during stopover events. So she did these trials where she exposed catbirds to either a rabbit, which is a non-predator, a Cooper's hawk, which is a recognized predator, or a cat, which was harnessed and in a cage, so it wasn't actually going to get the bird. And she wanted to know if the birds were recognizing the cat as a predator and if they were changing their behavior during these stopover events. So she put transmitters on them to see what, how they were behaving post release. So if anybody wants to tune in, I have the link to her Zoom meeting or defense. This is another PhD project where it was sampling fat from migrating songbirds to see if we can infer previous stopover location using stable isotopes on the extracted lipids from the fat samples. This is another PhD project that we're doing at Powdermill. This is looking at stopover behavior of habitat specialists versus generalists. And so this is using some transmitters with this array of nodes, which are receivers. And that's what the blue dots are on these maps around the banding station. So the birds get these transmitters and we can see what they're doing, how they're moving, how long they're staying during these stopover events. And we're also collecting fecal blood and feathers. I think we're not collecting blood from the birds that get, I don't know, transmitters, but we don't want to be too invasive with each bird. So we are going to have some idea about what they're eating on their stopover events as well. Soon, there is a postdoc who's going to be analyzing our banding data from the years that we have information about what nets the birds came out of. So for a long time, we didn't keep track of what nets the birds came out of, but this is maybe something to consider, numbering your nets, keeping track. So she's going to analyze the data to see what birds seem to be caught together. So this concept of co-migration, which birds are migrating together throughout the migration season, how are they interacting? And then there's going to be another student or maybe a postdoc who's going to ask the question, why are these species interacting? Why are they migrating together? There's other uses of banding data, maybe non-specific projects here, but this is non-exhaustive. There's so many things that we can do with banding data. The staff at Powdermill doesn't have the time to be able to do all of this. And so sometimes people have proposals, they want to use our data for some reason. They write a good proposal, they submit it to us, and we'll do a data use agreement. There's somebody who just requested our tufted titmouse data because he thinks that they're probably they have a eruptive migration, and so he wants to analyze what that looks like. Okay. So if we take a step sort of the other direction towards outreach and education, we also have a new classroom space. So this is part of the new building as well, and it's hard to capture this room in a photograph because you have to stand in the corner, but it has seating for up to about 25 or 30 people. It has a banding table that's not blocked by the field techs. They sit behind now. It has a TV where we can put a camera directly on the bird. We can show up close details of feathers or molt, or, depending on the audience, just showing people fat and talking about how birds migrate and what they use to fuel migration. But this is a really great outreach tool. So for both things like workshops and professional development, or kids, field trips, or just other sorts of groups that come visit. We have somebody who is, they want to quantify how well we as banders, most of us are not trained educators, we want to know, are we getting our message out there? So if we have groups, whether it's public or professionals or what, are we able to talk about what we know, teach them about migration, teach them about science, teach them about the importance of conservation? How are we doing this? If we're doing it well, they're going to figure that out. If we're not, they're going to figure out how can we do it better. Okay, so some of the things that we do at Powdermill for training, we do a lot of bird banding workshops. So in-person, hands-on workshops where we teach people from the basics of handling and extraction to aging and sexing, looking for molt limits, trying to recognize what is pointed versus broad when we're talking about feathers. We also do a lot of the North American Banding Council has certification sessions where we have space to be able to host that, and it's worked really well. Let me see what let's talk about. Okay, so we would like to expand that. So we want to do more intensive classes maybe. So before COVID, we had a grant where we were able to bring 13 Latin Americans to Powdermill to teach them an advanced ornithological techniques class. So we really did some heavy banding, we did some modus, and then we had people talking about things like the importance of citizen science. Somebody talked about bioacoustics and how to monitor populations that way. There were some metabarcoding people. So they did hands-on work, they did classroom work, they had a couple of field trips. This was great. At the end, 2 of them stayed on as interns through the fall season. They did a certification session, so they got their North American Banding Council certifications, and then they went home. So the idea is that our birds are not our birds, they're the hemisphere's birds. So if we can form these collaborations throughout that Western hemisphere, we're all going to be stronger banders. The birds that we band are going to head, well, some of them don't go south, but a lot of them do go south and then they come back. So we're able to have similar techniques across the whole hemisphere. This just makes us stronger as ornithologists. All right, so then there's the public outreach, just being able to bring people to the banding station, whether they're adults, learning about molt, whether they're kids practicing misnetting and banding, just seeing something up close. serving local brews. That does engage people. It makes them interested. It makes them want to come back. Okay. So I think just talking a little bit about how, you know, we really care about the research, and as banders, that's maybe, and maybe not for all of us, but that's maybe the primary thing we think about is gathering that scientific data, but also thinking about how we can teach others about what we're doing and why we're doing it. That's what's going to really boost conservation. All right, so I'm going to switch gears just a little bit because we got a grant to build a new building, but part of that grant was to build a data entry program. So if you're like me, you learn to band by writing your data on a piece of paper with a pen. But a few years ago, Powdermill went to just directly entering the data into the computer. And it was an old access program. It was fine, but it wasn't great. And so we were able to hire a software company to help us build a new data entry program. So it's quick. It really improves data accuracy and integrity, and I'll tell you exactly how. And it makes it a lot easier to submit the data once we're done banding. So it is a Microsoft Power app. This is where I'm breaking down a little bit. I'm not sure how these inner workings go. The data is stored on a cloud. The Carnegie Museum of Natural History, which is where we are part of, they're the tenant. And that's what I don't understand what that means. Luke DeGroat, he can tell you all about it. Mary, sorry to call you out, she's here. She might be able to tell you a little bit more about it as well. But let me show you how it works. So you can use this online or offline. You can use it on a computer, you can use it on your phone, you know, any kind of computer screen, you can use this. It either works in a browser or as an app. When you get your bands, you can enter the inventory. It keeps track of, you know, which bands are being used at which station. So we have 2 banding tables. If we put in which room we're in, it knows what inventory, what bands we would be using in each room. And so it automatically populates the next band in a series of, you know, you put size 1B, the band number pops up quickly. So another thing, this is something that I really like, we can attach photographs to a banding record. So we enter the data, I take a couple of quick pictures on my phone, I can log in on my phone and just upload the pictures right away. And so each banding record that you have a photograph, that's what it looks like. It also, we are able to store the minimum and maximum expected wing lengths, tarsus, mass, whether you should be able to age and sex a bird based on what month it is. And if you make a mistake, so either you're outside the range of that species, or you've measured something and it was totally different than the last person who measured that bird, it's going to come up with an error box. And it says, are you sure about that? Double check what you just did. So I did this mock black-capped chickadee with a wing length of 90. It's like, no, that's not right. And this is really great. So I'm going to keep going because the wood thrush saying, then you can visualize what you've done. So you can go back and it shows you what you've banded, how many, just make some really great graphics. So I can answer some questions about, you know, how you use it in the lab, maybe not the inner workings of it. Our goal is to make this available for any bander who wants to use it. We're in the process trying to figure out how that's going to work, but that's our goal. All right, so does anybody have any questions?
So questions for.
Oh.
What is your timeline for making that open?
So that's, I mean, that's a good question. We don't know. We're working on it with the software company. Yeah, the developers. Yep. Yeah.
When will?
I think that you'll know. We'll let people know for sure. Okay. All right. So there will be a small fee for the licensing. But
They're trying to work through. Shares, probably, the attendance. Lucas had.
Yeah. Yeah.
As far as like attaching photos, not a large file, text is small, but like it's data accessing and feeds, do we know how that works? Like space, like data storage, like if you attach a photo to, 1,000 birds banned every year, that's a lot of photos, that's a lot of data stored. Like what cost will that be to like maintain that?
I haven't uploaded that many pictures, if that helps. Yeah, it's just something. I tend to do it if it's like, all right, well, that's a really great molt limit, or this is a really good example of an aging or sexing criteria, or this is something really weird going on with this bird. This is unexpected. We might want to go back while we're proofing data and say, here's what I saw. Like, let's think about it a little bit more, because we want to let the bird go, not look at it for 15 minutes. So
As far as finding out, I would say, somebody had asked, just keep track of our Facebook page, because Lucas, he had a, like, online
he did.
about this to go, and so there's more development of what's there.
I should have put his email address on the presentation, but yeah, just reach out if you have any questions. Thanks, guys.
So many great takeaways from that talk, and I think one of them is that even big banding stations don't necessarily do everything alone. Collaborations are key. I think that's seen in a lot of talk as well. So even if you work at a small banding station, reach out to maybe people on council, the Powdermills, or other bigger banding stations, and see if there are ways that we can contribute and participate to those projects. So next up we have Bob Curry from Villanova. Wait, I got my thing back. And every talk about that I've ever attended, I've enjoyed. So the stakes are high. The one about using radio frequency identification and studying behavior in the ecology of Carolina black-capped chickadee and their hybrids.
Thank you. Glad to be here. So I'm a different kind of bander. 99.9% of the birds I've ever banded were chickens. Quite a few of them. We're up to 12,000 or so. But what I want to focus on today is one piece of sort of a methodological emphasis within our overall research program, which is, this will be our 25th year of trying to study hybridization in chickadees. We're focusing on, I'm going to focus on just one of the methods that we've used along the way to do a bunch of different things, and that is, if this works, no, it's not. That little thing. That is a pit tag, an RFID pit tag. So I'm going to focus on how we make use of this technology to study some of the details of, especially behavioral ecology of chickadees and even some chickens. Okay, so RFID is radio frequency identification, and it all starts with this pit tag. It's a little glass tube with electronics inside. It's a passive transponder, so the easiest way to think about it is it's easy pass for birds, because your easy pass does not send out a signal. It reads the antenna that reads the signal when you drive through the toll booths. It interacts with the magnetic or electronic field from the antenna and generates a unique code. So you have to get this onto the bird, or you inject it into your dog, because it's the same technology that vets use to chip your dog. So we're using leg bands. With larger birds, you could do it subcutaneously. We haven't ever tried doing that in chickadees, but you can do it with lots of other organisms, fish, sea urchins. We've done it for lots of different things. And there are other ways to attach it. You could super glue it to the back of the bird or something like that. But we've used leg bands. Then you need an antenna because the tag only works when the bird goes to where the antenna is. So that's a limitation of this methodology. So that could be a bird feeder, which is what we use mostly. It could also be the entrance to the nest, but people are using it to do other things where you, as long as you have a location where you know the bird is going to go, you can put the antenna there and hope to pick up the presence of the bird. So we've applied this in chickadees in lots of different ways. the chickadees, as everyone knows, are more or less permanent residents. We do get eruption visitors, and I'll say a little bit about them along the way. So there's one of the advantages of a permanently resident bird is they're here in the winter, so we can do winter work using bird feeders, having to do with their social ecology. And because we're really interested in the breeding biology of the chickadees as it relates to hybridization, we need to understand what's going on in the winter, because that's when a lot of the mate choice happens. So the social relationships in the winter almost certainly have a lot to do with what ends up as hybridization. I'm not going to say very much about hybridization today, but if I'm be around, if people want to ask questions about where we are with that, I'm just going to focus on this methodology. So our study sites are scattered from Great Marsh, which is below the area where there's overlap between the two species. So Rushton is about here, and Great Marsh is in Chester County, about probably 30 kilometers as the crow flies, or as the chickadee flies. And then Nolde Forest is a state park in Berks County, which when we started was in the hybrid zone, and now it's not, because the hybrid zone has moved north very quickly. Hawk Mountain was just barely at the edge of the hybrid zone, and now it's smack in the center of it. And Tuscarora State Park was outside the hybrid zone, and now we've got hybrids there. So the hybrid zone is moving north very quickly, and we've been tracking that all along. So we started in 1998, and the part I'm going to be focusing on with using RFID is we started in 2012. We did it for a couple of years, focusing on winter social ecology and breeding behavior. And now we're getting into, I'll get into the details about the dates. So we are still using this. But now we've started using it in some new ways to study cognitive ecology and communication in chickadees. So we started out with focus on the winter behavior using bird feeders, and most of our data came from outside the hybrid zone because we wanted to work it all out, starting with the Carolina chickadees to make sure that we knew what we were doing, and then the plan was to move up to the hybrid zone later. It's farther away from Villanova, so it takes more time to get up there to the hybrid zone, so we start with the local stuff. And initially, we used the methods that Dave Bonter at Cornell developed, where you take one of these little glass tags and you glue it onto the color bands. So we had 2 color bands. You have to actually encase the glass tag in plastic because you can't glue the glass, it'll let go. And so you end up with this combination, and then you put the antennae out there and you track whatever you're trying to track. So the way we did this is our Great Marsh site in Chester County. So this is all Carolina chickadees. Each of these green dots is a nest site. We use plastic tubes as essentially a nest box. And then the red and blue dots are feeders that have an antenna included. So this is a feeder. It's a plastic tube with seeds up in the upper part, and the electronics are embedded in the middle, along with the perch and the antennas right there. And this is at Nolde Forest. So these 2 projects happen simultaneously, where in both cases, we wanted to know which individual chickadees were going to which feeder and with which other chickadees, because a lot of this had to do with their social relationships amongst themselves. Okay, so we did this for a couple of years in 2012, 2013, and then we revisited it at Great Marsh in 2018 to see if the patterns differed between years. This involved a bunch of different students. So all of this requires that you catch the birds and band them with USGS tags, and we put color bands on too, but the color band at the time was attached to the pit tag. Then we start seeing who shows up at which bird feeder. So the way the system works is every time the bird lands on the antenna, it gives you a timestamp of the pit tag code. And then in our database, which is like the one we just heard about, it's now on the cloud and it's integrated with the USGS data and all the other data that we have. It tells us which bird it was. Okay, so that's the starting point. But then we want to figure out who is with whom. So this takes us into social network analysis. So what we're doing is figuring out there's a time series of visits at the feeder, and we want to know which birds come together. So if 2 birds come one right after the other, and they do that over and over again, then we assume that they're associated with each other. Could be male and female, but they're almost certainly part of the same flock. So there's software developed by Damien Farine and other social ecologists to come up with a group by individual matrix that tells you which clusters of birds included, or which clusters included which individuals. So the individual is either present or absent in each bunch of birds that came to the bird feeder. So this could be 5 minutes in between, it could be 10 minutes in between here. You get a flurry of visits at the feeder, they go away, and then another flurry of visits at the feeder. And each of those flurries is a group. And then you use some math to put that all together to see how distinct are the different groupings. Because it's quite flexible. The birds are coming and going, and there's overlap between flocks at a given feeder. There's differences. The same flock might come to one feeder and also at another feeder, but when they go to the other feeder, they run into other birds. So to put that all together, you have to do all of this comprehensively for all of your data. We're talking tens or hundreds of thousands of data points, and you end up with a social network map. So in a social network map or graph, each dot or node is a bird. I've color-coded these. That one is black-capped chickadees and Carolina chickadees, I think. So the dots are the birds. The lines connecting the dots are called edges. Those are measure them as being associated. And then the polygons are a secondary analysis of seeing who's clustered with whom. So it's essentially the different flocks. Okay, so I'm just going to give you some examples of what this looks like without worrying about the actual details of what it's telling us. But at Great Marsh, we are looking for the relationships among the Carolina chickadees. Now, if you remember the map I showed, we had bird feeders in 3 different sections of the site. So this group down here is a bunch of birds coming to one set of geographically separated feeders, and these are birds at the other 2 geographically separated clusters of bird feeders. So that's not telling us very much, but what this is telling us is there are 3 different flocks within this area and 3 different flocks within this area. In this graph, the red are the females and the blue are the males. So we can look at, oh, I'm sorry, no, this is blackcaps and Carolinas, because this was an eruption year. So in eruption years, we color band the blackcaps also. This is below the hybrid zone. We got visiting blackcaps, and we can see how they fit in. So one of the things we were interested in is, do the blackcap chickadees form their own flocks? And the answer is no, they don't. There's a bunch of different ways to measure that. One is called assortativity. You ask, are they distinct? Do you have blackcap flocks and Carolina flocks? And we didn't get that. We get the 2 sets of birds are mixed in together. So these are some more data showing this. So assortativity, if it's 0, means they're completely mixed together. And the measures we got are pretty close to 0, which means the blackcaps and Carolinas are integrated together in these flocks. They're not all in the same flock. So we've got a flock here with blackcaps and Carolinas, another flock with blackcaps and Carolinas. It shifts across the season. So the boundaries of the flocks and the home ranges shift a little bit over time, but the blackcaps stayed integrated. These blackcaps didn't move. They came down in October from wherever they come from. If someone can tell me where they come from, I'd love to know. We've never had a banded blackcap arrive already banded. We think they come from upstate New York or from Ontario or someplace like that. Once they get here in Pennsylvania, they don't budge at all. They sit for 3 months in the same area, integrated with the same local birds. So I think we've got the only data set that shows that for individually color banded and pit tagged blackcap chickadees. And then there are other measures we can do about all the different details going on in these flocks. So I'm just trying to give you a flavor of what we do using this system. So the second type of project we did, which actually involved one of the same students, Christy Eldridge, was to use the same technology in the spring at nests. So there's the antenna right at the entrance of the nest. The electronics are down at the bottom of the nest. And then you can keep track of what the mom and the dad are doing if they're already pit tagged. But you also get other events. So this is what chickadees around here have to deal with. That's an unpit tagged house wren dragging the pit tagged female chickadee out of the nest so he can then break the eggs and toss them and take over the nest. So that's a whole nother project someone could do, would be to pit tag the house wrens and use that as a way of studying the interaction between the two. It's not pretty. They're not our friends. But these are the kind of data we get. This is just descriptive data showing over the hours of the day. How do the visits of the nests play out? There are other ways to get these data, but the pit tags are really good. We don't know for sure that the bird was bringing food every time it visited, but we know that they were going in and out of the nest. We can also look at how long the female was in the nest between the time it went in and the time it went out, et cetera. And then we can correlate that with other data. Like, so this is the brood size across a whole bunch of different nests. This is 649 samples. So it's nest days. So we get about, back in those days, we had about 50 nests a year and we tracked them for several days. So we got a lot of data points. And this is just how the visitation by the mom and the dad varied as a function of the brood size. So the visitation rate per nestling, they didn't keep up. So that's a downside of having a large brood, is that each kid gets less food. So that's just an example of what you can do with this. So I want to talk a little bit about the methodology and the pros and cons. There are some downsides. The electronics sometimes are hard to keep running. If you get a short circuit on the board that reads the data, that's a problem. You could fry the board. If you get one broken wire anywhere, it stops working. And the batteries die, you have to keep batteries running, and you get animal damage. What do I mean by animal damage? These friends, okay? So if a squirrel bites the antenna once, it stops working. That's all it takes. And you can see the squirrel didn't just bite the antenna once. It thought this bird feeder was really good and chewed a hole in it. I didn't bring in a feeder that's been manipulated by the bears, but if the bear gets to it, it destroys the whole thing. And that happens a lot. Not at Chester County, but up at Hawk Mountain and Tuscarora, we get a lot of that. Some other downsides is some of the birds are invisible. If we never tag them, they're part of the social network and we're not seeing them. So we're only getting a biased sample of the data. The tags themselves sometimes stop working just for reasons we don't understand. They're intact, but they just stop. And sometimes they fall off. And the tags have caused problems for the birds. So that original system, the tags were kind of heavy and had sharp edges. Okay, so we stopped using them once we realized that was happening. And then we solved the problem. Okay, we didn't do it initially. This is a tag from a company called Excel, which dropped the mass down from 0.5 grams, which on a 10-gram chickadee is significant, down to 0.2 grams. The pit tag is here integrated in the color band. But that wasn't perfect. It's 60% lighter. That's great. Same tag. But we still got some injuries. That tag on a chickadee size band is 8.5 millimeters long. So if any buildup of keratin happens inside the band, it gets stuck on the foot and you can get foot loss. Some of those birds survive for a long time, but it's still, we want to avoid that at all costs. The problem has not happened in titmice. The titmice we've tagged, no problem at all. And in mountain chickadees that Vladimir Pravasudov studies in California, no problems. So it's extremely idiosyncratic. But then I solved a problem for our local birds. I took the pit tag and cut out the middle. And the problem went away. Because now the remaining plastic is very flexible, so that big long tube isn't a problem anymore. So I've had no foot loss since I started doing that. So I think that's the way to go. I just take an X-Acto knife and cut it out. The last thing I want to talk about is, running out of time, is our smart feeders. I have one here I can show later if anyone's interested. If I can run it. Got to do this. So this is a smart feeder. It's got electronics inside. And when I put the tag over the feeder, it opens up the door. Okay, so there's the pit tag system inside there. But in addition, there's a motor. So the motor controls whether the door opens and gives the bird access to the seeds. And we can program that to only allow certain birds to get access to the food. So we're doing this for two reasons. I didn't invent this. This was Eli Bridge and Vladimir Pravasudov invented this, but we've used it to study cognitive impairment in the hybrid chickadees. So the idea is, I'm not going to have time to go through the data, but if anyone wants to see some of the data we've got so far, the idea is that the hybrids are not, they have problems during development that affects their brain function. And for chickadees, what really matters is their spatial memory. So the way we do this is we have a frame with 8 of these feeders, and we assign each bird to only one of them. And then they have to figure out which is the right one. And what we're measuring is how long does it take them to figure that out? And cut to the chase, the hybrids are terrible at it. They do really badly. Okay, so this is, there's the data. So and we're also getting the social network data at the same time because the social relationships might confuse their ability to go to the right feeder. They might get pushed away and end up making a mistake, or they might follow somebody else to the wrong feeder because they're socially reliant or dependent. And lastly, so we do have some data about this. Sorry, the last project is a brand new one that I want to tell you about. It's using the smart feeders to study their communication. If a chickadee comes to an open feeder, what's he say? And then if he goes to another feeder and all of a sudden the feeder has closed because we programmed it to do that, does he communicate to his friends differently? In other words, are they telling their flock mates about whether there's food there or not? No one has ever studied that. So we're in the middle. That's a new study that we just started. So this system has a lot of opportunities, and there's as long as you get the safety aspects worked out, it's got a lot of potential, and there's lots of new questions that are possible using this. So with that, I'll stop it. We don't have time for questions. I'll be here. Okay.
So if we have questions.
Yeah.
The increased spatial abilities of the hybrids. So I'm assuming that these increased spatial, what does that say about numbers of people?
It's terrible. So at Hawk Mountain, the hatching success of the mixed pairs drops from 80–90% down to about 10%. So it's really bad to be paired up with a heterospecific, and kids that survive are the ones that we're studying, and they don't do very well. So our preliminary data suggests that they are not as good at the spatial memory as the unmixed birds. The first person to study that was Rice Lehigh in captive chickadees, and she got good data showing that the hybrids did poorly in captivity. We're trying to measure whether it's also true in the wild, and we can match that up with hybrid individuals have lower survival. So in mountain chickadees, the birds with there's variation in mountain chickadees with regard to who has good spatial memory, intraspecifically. And the individuals with bad spatial memory and mountain chickadees have lower survival. So that gives us reason to believe that the same connection exists for our chickadees. So it's not good to be a hybrid chickadee.
Maybe one more?
Yeah.
Check. So, have you ever seen on re-rates these divides and studies? I mean, learning on the
Improvements on read range. Not really. The technology, there's more than one frequency you can use. There's a couple different versions of RFID technology. And that matters. But the read range is within 10, 20, 50 centimeters. Okay. So they got to be sitting right on. For us, that's not a problem, but you can't use this to monitor birds wandering around in the woods away from a bird. Yeah, so it's not bonus. But for detailed stuff like we do, it's pretty good. The whole time we've been building. So at Hawk Mountain right now, almost every bird is a hybrid. And that has changed, because when we started, only 10% of the birds were hybrids.
Carolinas swept in and they were in that building forest. They were all hydrants that started and now they're all Carolinas. Yeah. Thank you, Andrea. This talk is only marketing about banning, but just an idea of how fair banning can be and to study things other than ornithology. And I should point out this project is basically the work of Spencer, who's the first author, he's a graduate student working with us. So I also want to point out that everything I do here is it's been approved for adherence to the Animal Welfare Act and all the other animal protection laws and regulations by various sites. You can see something in the line. Well, what is species disease? This is a fairly new emerging disease, which is hammering the beach, which is already suffering from beach spark disease, which was described in Lake County, Ohio in 2012. And at the time, it seemed to mostly affect sandwich, but turns out it affects older trees only slower. So the first symptoms look like this. Look at the leaves in the veins get opaque and stop producing energy plant. Eventually, the immediate symptoms they start discoloration and curling. The main symptoms, the leaves fill up and are essentially gone. And the families will survive for about 2 years, older trees longer. But this is the eventual endpoint of this. And so far, it's going to be 100% more trees on these trees that are affected. Not a good thing. The fish does American beech is extremely valuable for wildlife. It's a dominant tree in northern hardwoods, so mainly hard Massachusetts in northern hardwood forests. So what causes beech leaf disease? There's even pneumonia, but it's strongly associated with this microphobus nematode, which they might also not now. Which is found in 45 to 90% of leaves of infected trees, and after the leaves fall off, 90% of the buds, it's going to be the leaves of leaves, and it's quite difficult to wait through the buds. So this map shows the range from each by importance value, especially it's a seaboard, especially in the north and northern Pennsylvania where I was just going to beech leaf disease. Well, this is the range of the map of beech leaf disease so far. I'll submit here. But basically, it's starting here in northern Ohio, in the yellow area. It's really studying, then jumped over to eastern Western Kentucky. You see what it is slowly filling in. And nematodologists, what do we call people studying nematodes? So it's weird. Wondering how on earth the United States there's microscopic nematodes are only a few microns long. Don't go over, if you've ever seen nematode rays, they don't go over thin. And the answer is probably birds. The two potential methods of dispersal of anything, and the chemical range of ectozoochlorine, I think not it, which means it's mentioned to the external part of the tail of the bird or and those. And the two hypotheses of how it's being spread. I go to a well-known and big dispersion of a number of things besides these from food they eat, acorns, now it's often carp eggs, so feathers, and then some carp bread, which is invasive species, the main methods of carp, different types of brine shrimp are spread by getting stuck to the feathers, legs, or sandflies, and the hermits that feed in farmlands, shredded potato and soybeans. or universe. So things and choices. So there's a lot of precedent in great strategies. So one of the papers from my colleagues first read was Professor through the depth trap, you have to be able to survive the depth trap. So I think the question of volume in common in the environment, you can very massive concentration hydrochloric acid mimic sulfuric acid. You see, after 1 hour, after live being occurred, the one that we all learned on with protein deficiency, even after 24 hours, temperature number for live at 1.7 pH, a little more acidic than most other development. And the low type 2, which is still extremely acidic, I think lemon juice is pure. 35% is still alive at 24 hours. Based on what's known of the food. And it's a time, most things that goes to first, especially small song rates, it's a matter of. So obviously, it's potentially happening. So the study objectives were number one, people can accept this into each wild birds and species disease is random. And then the people detect the DNA and species of each wild birds that are fed and infected bees bugs. Although since Spencer made this program, eventually changed it from trying to feed the birds bees bugs, although a lot of birds eat bees bugs in the winter, making a bees bug solution. We created slurry of nematodes in water and just giving them a little drink solution. We had that to pass through the digest tract, and then finally to detect the nematode externally on wild birds and beach sheep disease areas. So to see which or both of these processes were actually happening. So the study sites were operating in northern Ohio, the lower right, and the Allegheny National Forest in northwestern Pennsylvania. Both areas have a lot of beach, high levels of beach sleep disease. We sampled in the winter 21 and 22, and then the summer and winter of 22, 23. The summer sampling was a simplified sampling. It's just to see if birds meddling around had the beach sleep disease nematode because it wouldn't be based on consuming beach sleep beach bugs because the nematode were running there during the summer. So the methods we use were basically established the feeders out in the woods. This is for the winter part. Get birds used to them, then spend the morning catching birds. And here's some birds we caught. Take samples for both fecal samples and other samples, and I'll go through how we did those. And run them for PCR. And PCR identifies the nematode DNA specific to this one species. And we're doing DNA partly because a lot of these are trying to identify a nematode, which is the nematode, because they all kind of look the same. Plus, it's a lot easier. So, in terms of collecting samples, when we catch a bird, hold in the bag for 50 minutes. Hopefully, it might sleep in that time and create a fecal sample before it's fed the beach bug and nematode mix. Then we brush it briefly with on the back half of the body with food grade diatomaceous earth. If you're not familiar with that, it's a skeletal remains of diatoms that are harmless, eating animal, but for microscopic organisms, cuts them into pieces. It's often used in the poultry industry to integrate persons. Then we brush that duck down onto paper and put it in a tube and use that as a sample to test for DNA. And then we hold them we give them the squirt of deep flurry of nematode water and put them in a cage with actual water and seed, deep bud milk until they produce a fecal sample. You don't know what happens if we then we let them And we ban them so that we don't take the same bird more than once. And it turns out birds really don't care if we hold them perfectly much, they usually do the go right back to the feed. Well, I think that they know where the net is and go over it. Birds are smart, except for the hybrids. So far, as of December 22, we've sampled a bunch of mostly chickadees, positional, for the hybrids, and a good variety of kinds of birds. 139, 21 species, over 200 samples of fecal and feather samples. Well, not feather, but feather dusting samples. And so far, we've had 10 positive detections of the nematode. And so I want to guess whether it's through the fecal route or the contact route. Fecal. It's actually both. Really? And it seems to vary by the month. You see the summer month here in August, we only had one positive. So one bird apparently had eaten beach bugs and becoming contact with beach nematodes. The actual increase in frequency is more an indication of perfection of our methods, so from the squirting the solution rather than feeding beach bugs. Because apparently a lot of things don't like beach bugs. I'm trying to, okay, so we have found nematodes in the feces of the bird before and after giving them beach bugs or beach bug juice. We found it in the eggs and feces. You can see here's a nematode. This is the fecal sample. You can see the eggs. And we have had birds, some preliminary work done at Holden Arboretum. I think birds naturally get seen beach bugs out there. So what's next? We're still not quite sure how the nematode is spread long distances by birds, because they come out of the bird really quickly. So I think the main mechanism for long distance first is but we're still not sure. Some of the birds that have been examined closely, the nematodes aren't actually on the bird, but on hippobostic flies that are on the birds. Another way that hippoboscs are horrible. It's unlikely a lot of birds that we're catching are actually spreading the nematode long distances, especially resident birds like chickadees and titmice and stuff. We're not sure. But once we figure this out, we can make predictive models for the future career. So not just for wildlife, but for people. And it'll be important to understand the consequences of beach loss, beach leaf disease and beach bird disease, the ecosystem functions, especially wildlife. A few things we're looking at. This is a part of that nematode solution. You can see all the nematodes in here. This is from like nematodes, and there are 1,000 nematodes wash out. Basically, you just soak the beach bud overnight in water, and the nematodes swim out to take a bath. What environmental factors might be affecting the spread of beach leaf disease and birds? What about beach nuts? They're an important mass, but are there nematodes in those too? We don't know. And what about caterpillars of beach leaves that have the nematodes on them? Do they take up the nematodes, and can they then spread them to birds? There's a whole lot more questions to be asked in the system. This is really kind of cutting edge stuff right here. I'm going to retire soon, so I'm not going to do it. So I'd just like to thank the people who would help with this. And if you have any questions, the person that asked is Spencer. He's the first to call from install. Thank you.
That's great. It's
Some other, we have discussed possibility of other organisms spreading it. But the forest pathologist and nematologist contacted us, myself and Spencer and some of the other co-authors who are ornithologists, so we'll naturally focus on birds. But it's another question that needs to be addressed.
Those positive samples you had, what species were there? The positive samples from the test, what species were the ones that actually had the?
Positive samples were mostly chickadees, titmice, and one other bird. I think it was a junco. Of the birds that they observed feeding naturally on beach buds, it was mostly titmice and chickadee. Any other questions? Or it's time for lunch?